I will continue the second half by talking a little bit more about structural biology rather than high throughput sex. So we're gonna mostly focusing in the next part about what we can do here in the second modularity on our beamline. That's the size exclusion coupled small scattering. In my short presentation, and Catherine, please interrupt me if I'm talking too long, okay? I will talk about a little bit about what we developed over the last, let's say, 10 years here at the SIBYLS, something about hardware and software, some application about size exclusion coupled SAXS and multi-angle light scattering. And I know Josh gonna mention in the second part of our workshop more about calculating structures or determining structures with MD, but I will give a little bit introduction how to use the data that coming from especially size exclusion coupled SAXS using Fox calculator and define crystalline structures. I think so you can see here is a perfect data for DSA, and with the beauty of SAXS versus cryo-EM as well was mentioned, we can use this constraint as well atomistic models, and we can calculate the data out of this. So you can see it's perfectly matched. Unfortunately, if you look closely at a small Q range, this region, that is where the misfit happened. You can do it in logarithmic scale, or you can also plot it in a more linear scale. You see with the large differences. And as you imagine, the DSA with bovine serum albumin have different oligomerization states. So if you're doing something in a batch mode, you see the signal from all those different oligomers. They're contributing strongly to the SAXS data. So you can imagine your sample is not only a fish in a tank, but a big fish or small fish together. Here's another example why we're supposed to use a separation method before we collecting SAXS. Here is another protein called ligase XSC4. And in a UV signal, what you're collecting at your lab, you probably see the nice single peak, and you think, let's head into the SAXS and collect some data. What you normally do. Unfortunately, when you collect the data for batch mode, the curve is the SAXS curve and the small angle are in the red, and the black is separated signal for just the peak. And when you do a Gini analysis that we didn't really mention too much about, but you can also calculate residue duration, and you see the different residue duration for this type of clean versus not clean sample. And you do also some calculation using the pair distribution function, and you see a dramatic change between clean and dirty. And then if you not really do separation on the size exclusion, you can do some. Okay.
Has it been 10 minutes?
Can we unmute everybody, please? So we can also do some shape reconstruction. If we don't like it here so much the shapes, but basically this is the first information what the structure may look like. You see a huge difference. So that's why we're supposed to do size exclusion if you do any structure modeling rather than just comparison of the scattering profiles. Here is a great example how similar can be scattering profiles. Those are shown in the P-raw function. John Tainer had a hand. Go ahead, John.
Sorry to interrupt. I was just looking at your ligase structure, the last slide. And I thought it was an interesting point about, the clean and dirty, but at the same time, I wonder, as you start to study systems where you have phase condensates or aggregation or things that may have a functional component, whether you want to actually analyze the dirty as well, or is that going to be really just a nightmare? Is there any way to get information out of that how heterogeneous do you think that area is going to be and so forth?
That's a good point. That's why I mentioned only when you want to do structure modeling. That means structure modeling of RNA, DNA, protein complexes that have one of these pairs homogeneous characteristic. If you go into these aggregates, that you study aggregates or condensate, you potentially not really do the structures that we showed the previous talk. There was a perfect example when the copolymers with the RNAs are basically aggregates or kind of conglomerates. So that's what is doing batch mode in high throughput. Here in this case, we can probably also separate the signal for this little bump here, that's aggregates. But it's, yeah, you can study that and just see the signal for the aggregates. That's definitely potentially possible. Normally what I'm showing here is just how to define, predict structures from the size exclusion coupled SEC-S.
Yeah, I guess you're getting at sort of nanoscale and mechanism and everything, but there may be this mesoscale assembly that worth keeping an eye on, since SAXS actually speaks to both, I guess.
Yeah. Some people really looking at aggregates and don't care. And I'll show some example when the complex state is much more complex and they really don't care about the monodisperse, or I would say the single species, rather they just looking on this larger stuff. I seen the project that they're really focusing on the aggregates right at the first.
Yeah, I guess my thought is that without the non-aggregated signal, the aggregate is kind of maybe less interpretable, but maybe with the structure of the individual assembly, whether you could then revisit that and get any other information. But it may be a diversion from your point, so sorry, just
no.
popped into my mind.
This is workshop, so we can chat and have this very discussion. Yeah, I kind of get this. I would just say, yeah, the aggregates or condensates start with a high throughput, and then if you want to clean up, what we want to do also with new separation methods, the FFF that we mentioned, could be possible to separate different aggregates, different sizes. Yeah, that's also kind of a new technique. What I want to this is a workshop, so I bring it up, how dangerous are these dirty samples if we do structure modeling. Here is just example when the red curve is just protein is dynamic versus the blue curve. That's when you have a theoretical data for some aggregates plus monodisperse monomer sample. And so they are very not identical, but they could be very similar. And the black curve is just the experimental PRR function for a non-SEC, so the batch mode. And so that's why, again, you can model it this way or model it that way. But if you do SEC, you can separate these aggregates easily. We developed at the SIBYLS size exclusion coupled SAXS and moles. You will see the tour during the lunch break. This is our fly tube detector, where here's the liquid handling robot what collected the high throughput data. I think Greg showed this slide, so I'm gonna skip this. But in the same time, we have a sample cell flow cell for our SEC-SAXS, where after the purification and the multi-element scattering, the protein go through the flow cell, and we can collect the SAXS as well. I give a credit to Daniel, who was incremental to really develop this in the last 3 years. Why I'm calling it autonomous size exclusion? We are not present during the Beamline collection at the Beamline. If everything's go well, right? If everything's go well, we don't need to be present there. It happens sometimes, depending on the sample quality, or sometimes users send us are really aggregates, mostly. We can take about 50 to 100 microliter sample. Concentration depend. If you have single peak, you can go to the lower concentration, but if you're expecting multiple peaks, you may expect at least 10 mg per ml. That's the highest what I try to see. It depends on the size as well. If you're looking at large molecules, you can come up with a lower concentration than 10, or can go to 10. So it's really questionable what the concentration. Sometimes uses another, they'll say 5 mg per mil, but they have a 7 peaks, and the peak of interest is probably like 0.2 mg per mil, but there's no signal. We're collecting size exclusion, then you have capillary, and then we also, after, we're collecting multi-angle light scattering. And now I want to talk about why we're using multi-angle light scattering together with SAXS. And this is very important. Again, we're collecting from plate. We can load it with multiple users. And as you can see on this example where you have the multi-angle light scattering show, that's this bottom line, show the molecular weight pretty stable across this very broad peak. But the radial duration that's coming from the SAXS is not stable. That's mean have different values. What it's mean that if we calculate the structure for this, it seems like this, and you see based on the SAXS, we find out that there is lots of movement between two domains, the brown one and the green one, but it's also a very transient or synergistic interaction between two parts of the molecule. And so that's why you can track this different compaction sometimes and extension across the elution. And that's why it's very important sometimes, if you're interested in this dynamicity, to distinguish oligomerization state differences versus dynamic state. Here is just, again, the same stuff. Structure that was crystallized, didn't fit the data at all. You can see the chi-square, the goodness of it is very high. And that's why we are doing most of the time this multi-state SAXS modeling. And I think Josh is going to talk about it a little bit more in details in the afternoon. So I don't want to go in detail. So instead, this is the crystal structure versus the solution state looks like this. And they have some other mutational work done to confirm this dynamicity. That word do not exist, but I like to use it. Well, the system is pretty complicated, and we build 8 pumps together with one autosampler. We can select different columns, different sizes. Everything is online. And switching between primary and secondary allowed us to equilibrate the system for next user and collect the first user. And that's why we are not needed to be at the beamline if everything's go well. Look at it. Yeah, well, we're still monitoring if everything's go well, even that everything's go well. Sometimes the evidence light source not go well, and our system works well, so it's back to it. 25 minutes per run, 30-minute purging for next user, and we can also do anaerobic conditions if there is request for it, but we like to have a heads up for this. It's a little more difficult process, and person need to come on site. Okay. Your data need to be online. You have a spreadsheet online that you fill up the name of the sample, what you're expecting, what size you're expecting, what column you want to choose. All those details online on our SEC-SAXS mail-in page. That is important because we automatically upload the spreadsheet for the users for the week, and we have ours develop some kind of a GUI or / script that is able to collect the data in synchronization with the moles, and that is then analyzed from us. You get the package, not only the SAXS data and the merge data, but you're getting also the moles analyzing in a PDF format. If you're requesting, you can get also the raw data or ASCII data for the moles. Please look at the moles before you go analyze your aggregates. Most tell you it's only aggregate. And even in the SAXS report that is sent it to you, you will get this double information that's aggregates only, or it's just exactly what you expect. So yeah, the data are deposited in a simple SAXS scattering. I would also recommend not only use it as a grabbing your data, but if you're publishing something from us, please go back to your QR code, the code of that you get it, and say, you have a permission to publish this data. out. So you as a user that take this data from SIBYLS can go back. I'm gonna publish this data. Everything's already there. You don't need to add there. You just kind of metadata if you're interested to add a little more details about what it is that the reviewers can grab it and analyze it by themselves. So that's why I'm showing this slide. Everything is ready for you, and I would like that the users are using this capability. Now, a little bit, this is a workshop, so it's a little training, okay? I'm not gonna do highlights only. If everything's go well, you have a separation of different oligomers and main peak. You get the similar for your moles. This is the SAXS, this is the moles. You can calculate molecular weight, you can calculate residual durations for the SAXS, but you can also get the molecular weight from the SAXS, check both of those. If you see something like this, we call the baseline shift. And the baseline shift happen, and the light source is not going well, our system is not perfectly tuned, or the buffer equilibration didn't finish properly. We may excuse ourselves, but it's not a big problem. We using, and your package, what you get is the data that are analyzed in a raw. And in a raw, you can go back and upload your HDF5 file. You have all the information in a README file, so you don't need to make a notes now. There is a possibility processing your data. We normally do this for you. You get your final curve, and you got the process and corrected baseline. But here is the baseline correction button that you can use if you want to reanalyze something. If you see something like this, the shift in a baseline. That's the first problem what sometimes may happen. It's not a big problem. The other problem is, this is again the baseline shift that show you when you take a buffer before and after. You may have a little bit different Guinea regions, but if you extend the Guinea region, you can cut the data as well. If you see something like this, you don't need the whole data sets, especially for smaller particles. You just cut the data sets. We have lots of questions for the users. My data is a small Q region, that's this regions are noisy. Yeah, it happens sometimes because your concentration is super low. So what do? You cut the data that are noisy and see if it's required those data sets for the Guinea or not. That's just a simple trick and tips. I would like to mention here, again, this is a workshop. Don't use BPS buffer, any phosphate buffer, please. Those are the worst. If you're using SEC-SAXS, don't use phosphate buffers. They cause the issue, we call it capillary falling. That means the aggregates are formed on our sample cell, and they contributing to this post peak shift. We call the baseline shift. This is a little bit post peak. It's not the same as previous slide, and that is also problem, but it could be also done corrected in a raw with baseline correction. But best solution, don't use the phosphate, use the HEPs or tris, or sometimes add a little bit glycerol in your buffer. Very simple. So here is example when you really go a little bit more complicated, and we have also a user here that is supposed to use this. I see right there that you use this deconvolution. And the beauty of SEC-SAXS, you have so much data sets across your peak. If you have a peak that are really overlapping, you would like to use this deconvolution method. It's called EFA. That is also part of the raw. And here's an example, and I'll show you how easily can be done it. You see there is some kind of different residual duration in the front of the peak, and then you have some fluctuation, and then you have something going on here, and there is a little peak here. So obviously this is very broad peak. Sometimes it is even more, like you can see a shoulder. You want to get as clean a sample, as small a disperse as possible. So you upload your HDF5 file that you receive from us. to so-called, and here, instead of LCS analysis, you can click on the EFA. And an EFA immediately show you what probability of components you have. And those 1, 2, 3 components are significant in this case. You define the range first. I'm going a little bit more educational here, but these are important points only. So you define the range. You can take the whole illusion. You can define the range a little smaller from, let's say, 100 to 400. And here it's defined how many components under these 2 peaks are. And it's a 3 components system. And so what you do, again, number of components, define, and then you hit next. And it's also show you where the components 1 start, where the components 2 is starting, where the components 3 is starting. These are the inflection points. I don't want to go in a theory on EFA, but it's defined from the back and from the front where the components are starting and coming in the signal. And then when you hit next, you can get final product, what is, you see that here is a little component at the front of the peak. That's the blue component number 1. There is a major component number 2. That's the basically main peak. And there's a little peak here at the end that is separated, but it's still, we have the whole range. So why not using it? And you see scattering profiles for all those 3 components. And in this case, because the little aggregates or whatever is this, higher oligomers, or as John mentioned, condensate, they're coming up first here and are overlapping our main peak. And so that's why you see how important it is to do the convolution, especially when you see some kind of residual duration is not stable across the peak. I would use it all the time, especially when you use this tool. It's pretty easy to navigate. And then you get even better signal to noise data if you do the EFA. Rather than for a few frames that you're merging, you can use the whole frame range, and it's averaging the entire component to is average of the entire peak. So those are kind of tricks and tips that I want to tell you if using SEC. Yes, there's a question. No? Okay. Application. And now we go to a little bit of science, what you can do with this. Okay, first I start with our COVID project. We all sitting at home. Daniel is collecting data with the mask on and completely covered because this is National Lab. We have him basically in a spacesuit at the Beamline. But we collect monoclonal antibodies against SARS-CoV-2 and against nucleocapsid. Gesundheit. And so the SEC-SAXS was very powerful in this case to show that there is a multiple peaks. If we have a different antibodies pairs, they are interacting with this nucleocapsid antigen. What is shown here is a pink. And antibody 1 and 2 are different colors, and the red one is the antibody 4, as an example. And from SEC-SAXS, you immediately see that, for example, antibody 1 by itself is eluding here. But when you're putting number 1 and 2 together, then it's the green light, the green trace. And the beauty of combining the molecular weight from the moles that is here in this line show you that if you combine 1 and 2 and 4 and 2, you form the same molecular weight, and there are this dimer of antibodies connected with the antigen. Antigen is very small in this case, about 50 kilodalton, versus antibodies are about 160 kilodalton. So you get the molecular weight is almost identical, and then you see the molecular weight. Fortunately, the horizon durations are very different for these 2 pairs of antibodies. What it tells us immediately that you have so-called networking versus a sandwich of conformation. We went farther and farther, and we find out that the network happens when the antibodies are rigid versus sandwich when this fabs can adjust and form a sandwich rather than network. I don't want to go in details here, but all those information could be done here with the SEC-SAXS and distinguishing conformation versus oligomerization. And here are just example of results for this sandwich-like versus network-like. This is very important for antigen test for SARS-CoV-2 in this case. When using the network-like antibody pair for your detection devices, you increase the detection limit for SARS-CoV-2 nucleocapsid. That is the main antigen used in SARS-CoV-2 antigen test. So that was kind of idea that we come up and started why it is important to use SEC-SAXS. And now I mentioned a project from Patrick Sheila by somebody here from Patrick Sheila. This is an older project with Albert. And he was also exploring SEC-SAXS on hundreds of samples, basically, coming every week. Because this autonomous SEC-SAXS, we can be not at the Beamline when he collect 20 samples overnight. That's feasible, even that we're running 25 minutes. And you can see this Rubisco-2 forms form a different oligomerization state across different species or species, hexamer, dimer, tetramer. He also studies some mutational experiments to disrupt these hexamers to dimer and see about how the kinetic parameters for Rubisco. If somebody don't know what the Rubisco is, the most abundant enzyme on the earth that take catalyze CO2 or take O2. Okay, so the simply that we collect the data, we merge the peak, and he was just doing a Fox analysis, basically take the predicted structure, existing structure, and feeding to the data, and see if it's monomer or dimer or tetramer or hexamer. Very simple. Not a big science, but due to this autonomous SEC, we've been able to do it. We developed this calculator that can take a structure and feed the data a long time ago. Here is a package called Fox. I will show a little example. I know that Joe's going to talk about BilboMD, but these are just basics. And Fox, what you can do with the Fox, you take your experimental data, and you take, let's say, a bunch of your models that you have from AlphaFold or in your mind or whatever models you have in the hand, and you zip those together and you put those here in this another button. There is also advanced options. You don't need to click on it, but there is like you can speed up the calculation if you're using residuals level. If you have some problems with the high Q feeding, you can click your offset, adjusting a little bit the offset, etc., etc. You can read about that in any kind of publication. Output, in this case, it's a small molecule, and you see there is different models that we have in the mind, and all of them feeding pretty poorly. Except the first one, it's kind of close. The goodness of fit is written here. Here are some parameters adjustable. They're adjusting automatically, and is radial duration of the model. And the goodness of fit tell you if it's good model or bad fit. And the chi-square is the lower the better. And so here chi-square 2 seems okay for the model, the red one. You kind of see it here that residuals are important to look at those rather than the fit. That sometimes it's hard to see if the fit is good or bad, but the residuals help you to navigate good or bad fit. There is a big button that is small, unfortunately, and I asked the web developer to make this button as big as possible. It's called multifox. When you click on a multifox, you can calculate so-called multi-states model, especially for this structure that we're expecting these two AGG-like domain may have some vibration. And that was shown here in this multifox model that the mix of this same structure in this case, but a slightly different tilt, a little bit, not too much, was required to match the data. Instead of 2, we got the chi-square 0.84. That's for 3 states model that fit the model the best. So how simply this goes, AlphaFold can produce now on the web lots of different models, or you have something already crystallized. I recommend still use AlphaFold because in crystallized structures, you're missing parts. You're missing your histoc, you're missing your loops. Here, if you do some prediction, they're gonna be almost the same in our resolution as your crystal structure, but it will add those histoc like you can see here, that matter a lot. Full structures are important for our modeling. And Fox is also part of the larger package, and BILBO-MD gonna be discussed by Josh in the afternoon after the lunch break. Here, just multi-Fox allowed you the calculation of multi-states, and FoxDoc has also allowed you to put 2 PDBs, 2 structures that you expect have a complex, and you have a data for the complex. You will arrange the complex to fit the data best, but it's also give you a docking fit. And those are all web applications that can be used. Here is just a little example, introduction to Josh's presentation. BILBO-MD calculated lots of confirmation, and it's all done under the app. So you don't need to do all these steps by itself. You enter your data and your PDB. It will calculate ensemble states, different conformations, and give you this kind of final output with a chi-square like this. And you see you need, in this case, this is a solo dehydrogenase that have a heme and a FAD domain. And obviously, one static structure fitted really poorly. When you add in the multiple states, you improve the fit from 2, what is single state versus 1 point something. What is the direct indication about dynamicity? I love using this word more. Dynamic structure of the cell BO's dehydrogenase. And this is my last slide. Okay, I'm doing good.
Yeah.
And then hopefully we're gonna start some questions. I can bring some more slides. This is again showing the size exclusion coupled SAXS and moles, where we tackled a really complicated system, the replication transcription complex of SARS-CoV-2. And what I want to show here, by just simple look at just the elution, we already see that there is some complexation when you put in NSP7A12 and RNA together, then it's the complex forming, versus when you have just the proteins, the non-structured protein of 7, 8, and 12, they afford some transient behavior with probably some partial complexation versus free states. As you can see here, the 7 is eluding the last one. It's smaller protein versus 12 is the RNAs. I think today is no, it's anyway, polymerase, RNA polymerase. And when you have a complex, it still have this transient behavior. And so by adding the RNA, the whole complex built together and formed this very sharp peak wherever you're using double-stranded or single-stranded RNA. And you can do this and get the final curves when you kind of see here by fitting those curves. And I'm fitting and showing here fit in a real space where the dot lines are theoretical versus the solid lines are experimental P-row functions. And so kind of we come up with that NSP 8 and 12 is in 1:1 ratio without the RNA. When it's RNA present, the 7 is coming into place and stabilize the complex with the RNA. So that's just what was confirmed also with cryo-EM, but they didn't have this transient kind of functionality of the RNA is or the NSP7 is not in a place all the time. It's just come in a place when the RNA is threading through the hole. And I think that's the last slide. Oh no, I have one more if I can.
You have a few minutes.
Yeah, okay, a few minutes. Okay, good. What do you do when you have the large stuff? And that's why we have a discussion about this large stuff. And when you purified something what is 10 plus megadalton on regular SEC, it's difficult. But you can use the different columns these days, or so full flow field fractionation that we ordered recently and coming. And so here you see like example for a collaboration with Cheryl Kerfeld, and this bacteria making the synthetic microcompartment. And there are different sizes and there are different genes responsible. But she found out, or her group found out, how to do those synthetics in vitro. And they find out based on EM that I'll show you later, they form the different structures, the different sizes of shells or tubes or octahedron shapes. And they've been interested which genes are responsible for which shapes and how you kind of change this assembly. And so we use SEC, a new column, forget the name, but it's a Wyatt column, SEC-1000 RecX, that is allowed to separate like 10 megadalton plus particles. And so we can see different samples at different elution time, or some in vivo expression of these BMCs have some very broad peak with some shoulders and tails. So this is just zooming in at the main elution, where here you have the single components, the non-complex state here at the end. So that's the beauty of this new technology that with the new columns. And when you look at those curves for these different peaks, and also we use on this EFA, the convolution for this broad peak, you get completely different curves. It also tell you the SAXS curves correspond to very different shapes, a very different organization of this PMC's large particles. And then you can go and analyze this, and you can show which is what. And I don't want to go here in details, but we're using so-called SLAP models, core shell cylinder or sphere, hollow sphere models. So those are not atomistic model, but one of the programs that we use called SASView, that you can use this to really fit this data with some model, a very primitive model, but this is what is the question. If it's a tube, or is it a sphere, or it's a very heterogenic tube. And you can see here, we are able to fit those data with different shapes and analyze what kind of sample form what. And I think that's my last, and I don't have a thank you to everybody, but basically the whole SIBYLS group is here. I think was shown by LJ, the whole group pictures. Okay.
We'll just pretend we're looking at Greg.
No, LJ showed this. Greg showed these slides, like the the SIBYLS group slides. Like, yeah. Anybody have a question?
I have a question.
Yeah.
Is there something else?
There are some hands up. Yeah.
Oh, sorry. The next to the last slide. So in the first peak, there's wide variation of RG.
Yes. So the RG are off
Is this
because the particles are so big.
The Guinea region is so small.
The Guinea regions are so small. So the raw. A good point. I mean,
this is something we see, and
yes.
it drives me crazy.
If you go into the aggregates and radio durations are like few points, you can rely on the radio duration across the elution peak. That's a good point. This is completely off. The radio durations are wrong.
Okay. Is there any way you would have to correct each slice, right?
Yes.
If you wanted to have a nice constant RG across the peak as you have in the second peak.
I would recommend merged few points only and look at the curve and calculate, the Guinea for each section manually. And if it's only few points, and this is also what we potentially will do in tutorials from 3 to 5, if you have that question, how do you define how much points is minimum for your linear region? Yes. Sometimes you can calculate the linear region. In this examples, what was shown for LMPs or other project that was shown as well, those linear region do not exist there. The nanometer size of those particles is 60 and 100 nanometer. You can get the linear region. Here's exactly the same problem. If you look at this, for example, this yellow one, that's this curve. This is in a log scale. That's not a residual duration. That's non-existent in this case.
Thank you.
I think, yes,
So I have a question about Fox.
Fox, yeah.
So let's say I have a single chain polymerase nanoparticle, and from the Cracking plot, it shows that it has some kind of flexibility or flexible region, but you can't tell for sure if it's just a mix of globular stuff and flexible stuff. So it's just a multi-domain kind of nanoparticle. Can I, first of all, is SEC-SAXS useful for the system? And also, can I use Fox to kind of
Yeah.
compute?
If you Fox is just, you already have some model in your mind. You mentioned it's a multi-chain or a multi-domain, or it's completely unfolded or something like that. That's the question. So I would definitely try just come up with some. It's a protein. I mean, it's a peptide or
It's a peptide.
peptide. Oh, that's problematic. Not problematic for the Fox. Fox can take anything. Any atoms can be taken in the Fox. Heavy atoms, peptoids, strange amino acids. But peptoids, I'm not sure if you can predict some kind of structure. You know, it's going to be some handmade models, you know. And you can come up with those 2 cases or 3 cases that you have in your mind. And if you can save it as a PDB, then you can zip it and put it in a Fox and see which models is right or closer. If you're gonna try something, what I recommend also, I can show during the tutorial, if you have your computer here and I can look at what kind of model you have in your mind, and talk about that. And potentially in the future, Greg didn't mention that, but we have office hour every Tuesday, especially for the users, our users. They get data from our Beamline, post-processing problems, modeling problems, or interpretation. Yeah, but we can talk during the tutorial.
Thank you.
So with AlphaFold 3, I've kind of observed some mixed results with its ability to predict oligomers. Like in some cases, it actually does quite well, but in some other runs I've tried, it just makes kind of really weird predictions for some oligomers. And I say that based on like comparison to some SAXS profiles that have indications for those oligomers. So I was kind of wondering if there's any discussion about implementing experimental SAXS curves with AlphaFold to kind of improve the robustness of like predicting oligomers.
Yeah, well, there is a big push. I think Susan is here also and maybe she was. I saw her on a Zoom. Yeah, we had some.
Yeah.
Yeah, well, somebody can respond that question better than that.
So we're trying to do it, but first, I don't think we can do it on oligomer. Well, first, we are focusing on just monomers and PDB. But basically, we want to provide extra information to AlphaFold to improve its prediction. However, the problem is that the limitation is still AlphaFold. It's like algorithm, for example, AlphaFold 3 doesn't focus on flexible regions and have worse prediction than AlphaFold 2. So if like the model's limitation is there, even though a SAXS curve can help them to get correct confirmations, it may not be significantly change everything. It's like we are trying to improve it based on experimental data, but not rewrite it or retrain. And still need some time before we can.
I also gonna mention, Josh gonna speak a little bit what we're developing now, validating the AlphaFold models, multimers or single species, single chains. He will talk about that, how we're using the PAA matrix to move the molecule. So sometimes you see discrepancy, all the AlphaFold models not fitting such data A, especially because it's not rigid or multimer is a little off, and plus some loops sticking outside. And so by adjusting this model, Josh gonna talk about that, we can fit the data and validate it, just validation, there is not new prediction. But just like, okay, this domain based on the PAA matrix, those are this confidence in fold and confidence in distances between the regions that we're using to fit the data better. Gonna talk about it. Okay, I
I
think.
think it's time for the next.
No,
speaker, and then
I think we,
we have
oh,
next-
Scott had the
Okay,
last
good,
one.
Scott.
So
Got
with
it.
this field flow fractionation device that you're purchasing.
Yeah.
What ranges of sizes of particles is it best for? Can you use it for really small 20 kilodalton proteins, or is it just for big things or everything?
For big and small too, we don't wanna use it for big, small, because SEC will probably give you better signal, there is less dilution. Will be a little bit more dilution for FFF. Good question. I guess on the company manuals or company kind of test cases, we can go from sizes of 100 nanometer, 1,000 angstrom, and even more, to sizes about 5 kilodalton. I mean, you don't wanna do the small one. No. But that is good for lipids. So that's the lipid protein assembly or lipid LNPs. And I think even for those copolymer RNA will be really good. That's why I should mention that we may use that. I think nobody tried this copolymer, what I think the lipid copolymer studies. Still hydrophobic, hydrophilic particle with RNA in it. So I think it's something similar from LNP. But that's where the fields go, LNPs and making cleaner data. But I think that's will be the main target, large, small LNPs. Okay, so who is next?